Okay, good afternoon, everybody. We're going to get this session started. This is the new tools in pancreatitis research session. Good afternoon, everybody. Hopefully you're having a great day so far. Yeah, got your attention now. Okay. Well, my name is Sobeida Cruz Monserrate. I'm an associate professor at The Ohio State University, and I'll have the pleasure to be moderating here, my co-moderator.
And I am Anna Gukovska. I'm the UCLA professor in the residence of medicine and VA senior research career scientist. And we will do it together today.
Yeah, so this afternoon, we have the pleasure to introduce Dr. Tamil Turks from the Indiana University College of Medicine. He'll be talking to us about imaging in pancreatitis, clinical PET probes, MRI, and AI. Welcome.
Thank you for inviting me, everybody. Good afternoon. So my name is Tamil Turks. I'm a radiologist at Indiana University. I have specific interest in the pancreatic MRI imaging and generating new imaging biomarkers for about 10 years or so. And first of all, is there any radiologists in the room? Please raise your hand. No radiologists. Okay. Okay, so I can talk whatever I want then. So we'll talk about new MR imaging biomarkers. Start with chronic pancreatitis, and then we'll switch to acute pancreatitis. And first thing that we need to know is about the PROCEED study, which has been going on for about 8 years now. It's funded by the NIH and as part of the consortium for the study of the chronic pancreatitis, diabetes, and pancreatic cancer, shortly called CPTPC. The PROCEED study is a longitudinal research cohort, and it has established that with about more than 1,700 patients now, and it still continues to recruit patients up to December 2023. And the aims are to estimate the risk of progression in chronic pancreatitis and define the risk factors and discover biomarkers for chronic pancreatitis, diagnosis and prognosis, and combine all these, the imaging biomarkers and framework, including biomarker genetics and mechanistic studies, and clinical information to establish a biorepository for chronic pancreatitis patients. One of the things that I've done in this consortium was to look into the MRI studies as part of the or an ancillary study for the PROCEED. And out of 1,163 MRIs, we collected 820 MRIs and published the results. One of the things that I want to talk about is T1 score, also known as T1 signal intensity ratio. And what is T1 score is, it can be very simply summarized on these two images. On the left is a normal pancreas. If you notice, the pancreas on this image is very bright, and this is not a contrast-enhanced image. This is without contrast-enhanced. The normal pancreas shows a very bright signal on T1 weighted images. In a chronic pancreatitis patient, we lose this signal. So that's what we are utilizing, and we are taking the ratio of the pancreas to the spleen, hence the name called signal intensity ratio. So we published these results in a new staging of severity of the chronic pancreatitis, and called it mechanistic stages. The difference and the beauty of the mechanistic staging is that it includes the Cambridge score within it, and clinical features determined by the PROCEED study. G2, Y1, Y2, Y3 are the PROCEED cohorts. And then also we include a calcification seen on CT. And as you can see on the right column, the T1 intensity ratio, or T1 score, was progressively declining as the disease progresses. This is the graphical representation of the severity of the chronic pancreatitis. There is a clear-cut, obvious linear trend of the T1 signal intensity ratio as the disease progresses. We published our results. You may feel free to go and read the full findings of that. Second study is an ancillary study, again within the CPTPC, called the Minimap study. There were 7 clinical centers and the data analysis center, which is MD Anderson. In this study, we looked at even more imaging biomarkers to evaluate the chronic pancreatitis. These included mostly quantitative MRI as well as non-quantitative findings. I don't know if you ever heard about the T1 mapping and extracellular volume fraction, diffusion-weighted imaging, arteriovenous ratio. And these are quantitative MRI, which are new imaging, the biomarkers. They are not for interpretation, but measuring signal obtained from the pancreas. So it kind of provides an objective way of assessing the pancreas. Other things that we'll look is visceral fat distribution, which has been linked with chronic pancreatitis, diabetes, and PDAC. T1 Dixon, it measures the pancreatic fat, as well as the liver fat in any tissue. And secretary enhanced MRCP was also performed. We are thankful to the Cairo Clinic for providing free secretin for us. And we measured the 3D volume of the pancreas and AP diameter. We also use AI to measure the pancreatic volume and classification of the pancreas. So what does T1 mapping look like? The one on the image on the left is a T2-weighted image, and the one on the right is also T2-weighted image, but in a chronic pancreatitis patient. As you can see, T2 is anatomical image. It tells you, shows you where the pancreas is, but it doesn't give you functional information and not much of detail. When you do T1 mapping, it basically gives you the tissue property, and which can be depicted with color. And the image on the left shows blue, which is lower T1 relaxation times. Patients with the chronic pancreatitis have higher T1 relaxation times. And we looked into the control and chronic pancreatitis patients. There was higher T1 relaxation time in patients with chronic pancreatitis. Second thing that we looked at was extracellular volume fraction, which is more commonly used in myocardial fibrosis. And it's, in fact, the standard of practice now for assessing the myocardial fibrosis. We use basically the same imaging technique to measure the amount of extracellular fraction of the pancreas. And how does that help us? Needs to go back to the normal histology of the pancreas. Pancreas is a very packed exocrine and endocrine organ full of acinar cells, and also that has a ductal, the tree. When chronic pancreatitis is fibrotic, there's increasing number of fibrosis and fat within the pancreas. And these white blob spots are fat, and the pink parts are the fibrosis. So amount of fibrosis increases in most of the chronic pancreatitis patients. Amount of fibrosis, as the amount of fibrosis increases, interstitial space increases as well. So we are able to measure this increase in interstitial space by MRI and using pre and post contrast T1 map and calculate the amount of signal within the tissue and also in the plasma, put it into a simple formula, and we generate the extracellular volume fraction. Result is, again, can be depicted in colors. The blue numbers on the left belongs to a Cambridge 0 patient. Red numbers on the right belongs to a chronic pancreatitis patient. So basically, as a result, there is a significant difference in normals and chronic pancreatitis patients in terms of extracellular or interstitial space. Dixon MRI is the technique used to measure the pancreatic fat. MRI is a sophisticated tool that can tell you how much of signal is coming from the fat protons versus water protons. And it can split that and show you amounts of signal coming from the fat, amount of signal coming from the water. If you measure the pancreatic signal on these and take the ratio, you obtain a pancreatic fat fraction, basically. And in chronic pancreatitis patients, we observed that there is significant increase in the amount of fat fraction compared to the controls. Then diffusion-weighted images did not help to differentiate the chronic pancreatitis. We therefore excluded the diffusion and combined these three quantitative MR features to generate a quantitative MRI or QMRI score. Having a composite score actually increased our statistical significance and widen the gap between the controls and chronic pancreatitis patients. Then we adjusted these results for confounding variables, and then the QMRI score after the adjustment for the confounding variables had an area under the curve of 0.75. Published our results. You feel free to go and read the full study, including the full details. What is our aim in the future? We are going to publish more studies. We will include more imaging biomarkers and generate a table with numbers underneath it so that we can assess the chronic pancreatitis severity and diagnosis more objectively. The second is a new consortium called Type 1 Diabetes and Acute Pancreatitis. And this has also a longitudinal study called DREAM study. And there is an MRI arm within the DREAM study, including 10 clinical centers. In this study, we will do longitudinal MRI together with the metabolic testing over 3 months, 12 months, 24, and 33 months. And these are, again, the imaging biomarkers that we are going to evaluate. Includes T1 mapping, signal intensity, ECV, DCE, diffusion. And one of the things that we'll look is pancreatic perfusion. Pancreatic perfusion has been shown to be increasing threefold during the islet self hyperfunction. We will look if this is disturbed in patients with acute pancreatitis. And then basically the perfusion or DCE images look like this. This is a prostate, which we commonly use DCE for. You can generate different types of enhancement patterns using the DCE imaging. Another thing that we will look is tissue diffusion. And in diffusion, molecules are free in normal state, whereas in inflamed tissues, there's decreased diffusion. This results in difference in the signal that we obtain from the inflamed tissues. And then you can generate images that looks like this and measure the amount of diffusion restriction. Another thing that we will measure is pancreas volume. As you know, pancreas volume is reduced in patients with diabetes. We will use both manual segmentation as well as artificial intelligence to measure the pancreas volume and correlate this with development of diabetes. AI can also do texture analysis, look into the tissue heterogeneity and see if this changes in post-acute pancreatitis patients, and will this predict the patients who will develop diabetes or not. Pancreatic fat, again, is also important in diabetes as well as chronic pancreatitis. We will measure the pancreatic fat fraction and determine how does it change. Another thing that we will look and we can measure and assess is sarcopenia, which is the skeletal muscle mass. This is easy to measure using post-image processing. The image on the top has much less muscle volume compared to the image at the bottom. Briefly, I can tell you that there's a contrast for ultrasound now, and it has been approved by FDA in 2016. Ultrasound is a flexible and practical tool if you know how to use it, and it can improve the characterization of the lesions seen on grayscale ultrasound. And it can look into the perfusion of the pancreas. It has been shown to correlate with the CT in terms of whether this is a necrotizing pancreatitis or not. And it can also be used to differentiate focal mass-like focal pancreatitis from adenocarcinoma. But the studies are quite preliminary right now. And F-flucyclovine PET can be used in autoimmune pancreatitis to differentiate the AIP from pancreatic cancer as well. But these are very limited and preliminary studies or even case reports so far. More studies are needed for contrast-enhanced ultrasound and PET. So in summary, CPTBC has been around for almost 8+ years and began publishing new MR imaging biomarkers for chronic pancreatitis. Type 1 DAPC, established about 2 years ago, aims to generate imaging biomarkers for post-acute pancreatitis diabetes. Thank you very much. Almost finished.
Excellent. Thank you so much for that great talk. Our next speaker.
Okay, so next speaker is Bomi Lee. from Stanford University, and her talk will be inflammatory cell profiling in pancreatitis.
Thank you for introduction, Anna. Thank you for inviting me for this talk. So basically, I'll be talking about inflammatory cell profiling in pancreatitis. So some of you may heard of this carousel of progress or have watched this show. Basically, this is at the Magic Kingdom of the Disney World, right outside of this hotel. And this carousel of progress is basically talking about the technological advances of the typical American homes, showing that in the past and current and future. So very similar to this, I prepared this carousel of immune technique progress. So I'll be basically talking about the immune techniques we have used for the pancreatitis research in the past. And I will discuss their limitations and difficulties, followed by current advances in the pancreatitis research, mainly using my research data as an example. And then I will be introducing some of the techniques which can further advance pancreatitis research in the future. So immune cell analysis of pancreatitis in the past has been performed by using the mainly histological methods, immunohistochemistry or in situ hybridization of the pancreatic tissues from the donors, or conventional immune single cell analysis, flow cytometry analysis using the circulating immune cells, PBMCs. This is mainly due to the inaccessibility to the pancreatic tissues from the pancreatitis patients. So we also, that's why we use animal models. And there's advantages using these animal models. First, we can easily manipulate genetics. And also it's easier to control the disease progression and then disease severity. However, we still need clinical samples to find the human disease-specific some immune signals. So I personally think one of the recent advances is use of TPIT tissue samples. So in collaboration with Dr. Melina Berlin's group, we had the opportunity to receive these exocrine pancreatic tissues from the chronic pancreatitis patients who underwent TPIT procedure. So as you can see here, we were able to uncover this, observe these CD45 positive immune cells in these fibrotic intact exocrine tissue samples. And by using the flow cytometry analysis, we further identify different composition of immune subsets in hereditary CP versus idiopathic CP. So overall, by using these TPIT tissue samples, we were able to uncover distinct local immune responses in the two different subtypes of chronic pancreatitis. And one of the recent advances in immune techniques is CyTOF, so which is also called mass cytometry. And it's pioneered at Stanford and which use metal-labeled antibodies. Which, technically, we can detect up to 100 different markers, but normally we can detect 40 to 50 markers. And then which allows in-depth immune cell profiling and identification of novel cell type within the antibodies on the panel. So recently, by using this CyTOF technique, the Murli Manohar from our lab identified novel circulating and tissue-infiltrated monocyte macrophages from the acute pancreatitis and severe acute pancreatitis mouse models. And also he identified distinct circulating monocytes from the blood sample from the acute pancreatitis and recurrent acute pancreatitis patients. So another recent progress is in-depth immune analysis is single cell RNA sequencing. So we use this single cell RNA sequencing is actually widely used in the different disease settings, but we use this technique with this TPIT tissue samples, and we are focused this immune cell profiling. So we've observed this different, identify different T cell subsets. And interestingly, we found distinct T cell, the gene expression pattern by the group, the control hereditary CP and idiopathic CP. And using this single cell RNA sequencing, not only we can identify disease-specific novel cell types, but also we can identify specific immune signals such as CCR6 and CCR20, which were significantly upregulated in the hereditary pancreatitis samples compared to idiopathic CP. So by using this technique, but actually we also run when we perform single cell RNA sequencing, we also perform TCR sequencing as well. TCR stands for T cell receptor, which allows simultaneous detection of transcriptome and also TCR sequences in the same cell at the single cell level. So TCR sequences serve as a barcode for the T-cell clones and key readout of T-cell identity and function, and which determine T-cell antigen specificity. By using this single-cell RNA TCR sequencing, what we found is there is significant alteration of T-cell repertoire, especially hereditary pancreatitis samples compared to control or idiopathic CP. This was mainly due to the distinct T-cell clonal expansion pattern, CD4 T cells versus CD8 T cells, and then significantly increased the infiltration of CD4 T cells in hereditary CP contributed to these changes. And furthermore, we also used TCR clustering analysis, and we identified these antigen binding motifs, mainly shared by these hereditary CP individuals. So overall, by using this single cell RNA TCR sequencing, we were able to uncover disease-specific TCR antigen responses. And we also established a codex, which is spatial multiplex cytometric imaging analysis. So which basically use 3-color fluorescence microscopy with this automated staining washing machine. And basically, this codex iteratively visualizes these antibody binding events using the DNA barcodes and in situ polymerization-based techniques. And which allows multi-cycle reaction of 3-marker staining up to 22 cycles, so we can detect up to 66 antigens in the same tissue slice. By using this codex analysis, not only we can identify novel cell types and their spatial localization, but also quantitative cell-to-cell interaction, and which allows a broader characterization of tissue architecture. And then we use this codex analysis using this hereditary chronic pancreatitis tissue samples. And as you can see here, the immune cells, epithelial cells, and pancreatic stellate cells are located in close proximity, meaning they are close interactions. So now we move on to future technology. So future technology has to be multi-omics approach, such as spatial TCR sequencing. So this spatial TCR sequencing combines spatial gene expression and then T-cell receptor sequencing, and which determine the spatial organization of T-cell repertoire and then visualize the localization of specific T-cell clones. So it's especially this technique is very useful to track the specific T cell clones and their interaction with surrounding cells, either immune cells or non-immune cells, and providing this antigen exposure potential and antigen specificity in this dynamic tissue microenvironment. So I think the future immunological technique also has to be 3D multi-dimensional approach, such as 3D projection of the optical clearing. So during the recent decade, there are many publications released regarding the 3D projection of the pancreatic tissues using the different optical clearing methods. And one of the advantages of using this 3D projection with optical clearing is maintaining the native hydrophilicity of the tissue. So we can also avoid artifacts produced by microtome slicing and dewaxing steps from the conventional histological method. For example, you can see here this we can clearly visualize these hydrophobic fat and adipocytes in these human pancreatic tissues. And we can also better visualize delicate lymphatic vessels and T cell infiltration. And people already have shown the feasibility of this 3D projection of the exocrine pancreatic cells by using the in vitro culture system. So far, I have shown some immunological techniques, mainly for the immune cell profiling from the isolated from the tissues or blood. But for the small molecules such as chemokines or cytokines, there are recent advances such as Olink assay or SOMAscan. In summary, technology and in-depth analysis tools have overcome previous limitations and advanced pancreatitis research. And current technology still has caveats and also limitations. Further validation and functional studies are needed. For this, we still need a conventional staining imaging approach. And future study require multidimensional integrative approaches to decipher the interaction between immune cells and non-immune pancreatic cells through the collaboration and then teamwork. Thank you for the attention.
Thank you very much. Great talk. Lots of new advances in immunology for us to advance the field forward. Next, we have Dr. Guy Grabowski from the University of Wisconsin-Madison. He's going to talk to us about novel cell biological techniques. And I think this is one of the few investigators still doing some of the amazing, carrying that legacy and training future investigators in the field of pancreatitis. Thank you so much for coming.
Should I touch this? Here. And how do I go backwards? I'll start here. Oops. Okay, thank you very much to the organizers for asking me to give a talk on tools in pancreatic research. I wasn't sure what to give a talk on. So I thought I'd tell you a story today about the characterization of exosomes or small extracellular vesicles that are released from pancreatic acinar cells. My lab is very interested in understanding the molecular control of membrane trafficking events in acinar cells, just the basic science of it. And so in this slide, you see the classic secretory pathway, immature secretory granules butt off with a Golgi apparatus, and then they condense down by losing membrane and some of the digestive enzymes into the endosomal system. And you can see they go into the early endosomes, late endosomes, and recycling endosomes. But the granules themselves then mature and condense down into mature zymogen granules, and they're stored until you eat. We spent a number of years characterizing this endosomal system, which was originally identified as the constitutive-like and minor regulated pathways many, many years ago, People knew it existed and it released small amounts of newly synthesized digestive enzymes, but nobody knew the compartments that were responsible for this trafficking event. One of the very interesting things we found, you can see that classic granules are shown in blue with vesicle-associated membrane protein 2, which is a snare protein necessary for fusion with the plasma membrane. What we found is that this constitutive pathway was absolutely essential to form a different population of granules, and these are decorated with VAMP8, which is an endosomal snare protein. And if this constitutive-like or minor regulated pathway was not intact, then you would not get the VAMP8 granules. Very puzzling. On the right-hand side is just a schematic of how secretion how digestive enzymes are released from a single batch of acinar cells. You get a quick spike immediately after CCK stimulation, followed by a second phase. The first phase, the immediate phase, is mediated by VAMP2, and the second phase is mediated by VAMP8. They both contribute about 50% of the secretory response, it turns out. So this all started because of a protein that we identified called tumor protein D52. I'm not going to tell you a lot about the biochemistry or the cell biology of it today, except that I say that it's a soluble cytosolic protein. It's not in zymogen granules. It doesn't have a signal sequence. It's highly expressed in breast cancers, hence the name tumor protein. We've delineated that it's an autophagy receptor protein that'll drag various molecules and phospholipids into the autophagy pathway. It undergoes phosphorylation. It's highly expressed in exocrine cells with large secretory granules, exocrine cells that release protein. So early on, we had to figure out a way to study this constitutive-like pathway, which we also call endolysosomal secretion, how we can look at it separately from zymogen granules. So we did what's called cell surface labeling, where you leave the cells intact and then you just immunostain for externalized epitopes of the zymogen granule protein synaptotagmin 1, shown in whatever that color is, purple, or LAMP1, which is not on the zymogen granules. You can see that on the blot on the left. And then we stimulate with secretagogues, and you can see that there's some overlap between the two pathways, but there's clearly distinct sites where this exocytosis occurs. Is this all relevant to pancreatitis? Yes, it is. Basically, in this slide, I'm showing you that we found if we inhibit trafficking in the endosomal system, especially trafficking towards the late endosome and lysosome, it completely abolishes intracellular trypsin activation in response to CCK, or not shown here, also bile acid. So this is the YM compound on the top left, where it inhibits an enzyme called PIK-FYVE. It's a lipid phosphorylating enzyme that's essential for trafficking. You can see increasing concentration shuts down secretion or shuts down trypsin activation. On the right, we did it by using molecular techniques, by overexpressing a wild type or a dominant negative. And underneath, the bottom two panels just show you that if you use a VAMP8 knockout, and this is the same VAMP8 that's on those granules, but it's also in the endosomal system. If you use a VAMP8 knockout, you get no trypsin activation. It doesn't occur in response to CCK. On the right-hand side, you don't get any LDH release either. So that it abolishes damage. I told you before that this D52 molecule seems to be involved in autophagy. And indeed, if you look on the left, those small dots inside those autophagic structures, immunolabeling of D52 that wound up in the autophagy pathway. On the right-hand side is staining of D52 with LC3, and they overlap. And so it's in the autophagy pathway. And so this really begins my talk. Serendipitously, I will say, it's a long story. My technician told me that D52 is in pancreatic juice, to which I said, no, it's not. It can't be. It controls secretion. There's a million reasons why it can't be. And so she got mad and collected pancreatic juice from rat, anesthetized rat, stimulated with secretin and CCK, used 3 separate antibodies and said, oh, yes, it is. And she was right. It is released, much to my amazement. We also looked in cancer cells, which release lots of exosomes. And so we purified exosomes. These are extracellular vesicles that are released from cancer cells. It's released from all cells, really. And we found that in cells that were stimulated with ionomycin, there was a huge increase in D5-2 release, and it co-purified on the exosome. CD63 is an exosomal marker. It's a tetraspanin on the bottom. We also isolated exosomes from acinar cells, and this was a tricky part. So if there's any tools here, basically you can't do it in BSA. The cells are very sticky. So you have to let them lay down on plastic, and you have to be very, very careful when you remove the media, not to bring any cell or cell debris up, and then you've got to spin it out. And it was quite laborious. We did this mainly because I wanted to do proteomics on these interesting exosomes. Exosomes from pancreatic juice are very difficult. Anyway, you can see D5-2 is highly, highly enriched in the exosomal fraction. However, underneath it, you can see calreticulin, which is not in risk, is in fact there. So these are not super pure fractions like we need to do proteomics. This is just a slide that shows you that exosomes come from late endosomes or multivesicular bodies, and they fuse with the plasma membrane and then are released into the media. There's another pathway called secretory autophagy, is shown on the right, where basically this autophagosome that forms can either fuse with a lysosome and undergo degradation, or it can fuse with a late endosome, just the multivesicular body, form what's called an amphosome, and can be released into the extracellular medium. We think that's what's going on here. One thing that's very unusual is people that study exosomes will take cells and they'll collect media after an hour. After a day or two to get enough to measure. We can do it in 30 minutes from the acinar cells if we stimulate them. It's extremely robust. These are the exosomes that come out of the acinar cells. On the left-hand side, we get two populations. Those with a diameter of about 50 nanometers, and those with a diameter of about 100 to 200 nanometers are larger. I won't go into a lot of detail here in the interest of time, but we also do dynamic light scattering to look at them. So then we do this whole litany of typical acinar cell secretory studies. So here you're going to look at D52 released under basal conditions, and you can see on the left, it's nice. It comes up at 15 minutes and plateaus. On the right-hand side, that's amylase from the same cells. This is with D5-2 secretion. The dashed line represents basal. So it's all normalized to basal. It comes out in response to low and high dose CCK, but you can see the error bars are pretty big here. This is where we're starting to learn how to do it. We need to go back and revisit it. Anyway, the cyclic AMP on its own discharges the pathway, independent of CCK, and that was predicted many years ago by David Castle using radio labeling. On the right side is amylase release again. So I'm going to skip this one. And so for the last section, I'm just going to say that early endosomes on the left-hand side on the bottom give rise to late endosomes multivesicular bodies. They then confuse with the autophagosome to form the amphosome, which can undergo secretion, or the autophagosome, the amphosome confuse with the lysosome to become an autolysosome, and then there's degradation. So this can be inhibited in a number of steps. If you use the drug I showed you for PIK-FYVE, epilimod, you'll block an early step. If you use a drug that is a syntaxin 17 inhibitor, it will block the formation of the autolysosome. And this is well characterized now. And if you use bephyllomycin A, you'll get the autolysosome, but it won't degrade. And so what we found, much to my surprise, was that here you see again, this is fold over basal D5-2 release. CCK causes a nice eightfold, you see the error bars are tighter here, eightfold increase in exosome release. We put in epilimod, it matches it independent of CCK. Bephyllomycin, not so much because you've already formed the autolysosome. The autolysosome. It's way down at the end of the pathway, but when we put in the EACC compound, which freezes the amphosome, sure enough, we got a huge increase in discharge. And so this was over 45 minutes, which is quite remarkable because other people that have shown this, it takes up to a day or two in order to get release of the exosomal fraction. Okay, I'm going to skip that. So just to summarize in these last couple seconds, D52 is secreted by an endolysosomal pathway in the acinar cells. It also occurs in cancer cells. It also occurs in pancreatic juice. It co-purifies and is highly enriched in exosomal fractions. Inhibition of autophagy increases the secretion of the exosomes into the extracellular space. And I'll just jump to the bottom. If you inhibit syntaxin 17, which really enhances the amphisomes, and you'll see a huge increase. It's a remarkably robust pathway. We never expected it. Can it be used as a diagnostic marker, et cetera? I hope so. We're going to give it a shot. Thank you.
Okay, so next speaker is Kevin Pavelka from Mayo Clinic, and his talk is single cell omics or other omics, spatial profiling or other big data.
First of all, I'd just like to thank the organizers for giving me the opportunity to present today. I'm a immunologist in a core laboratory. I'm not necessarily, don't necessarily directly do pancreatic research, but I study the immune system. And as evidenced by several talks and posters, the immune system is important in pancreatitis. So I think some of the techniques that we're using in the core will be readily applied to your pancreatic research. So, and so our core focuses on high dimensional single cell analysis. And so we use, single cell proteomics, and the idea is that we want to be able to analyze whole blood cells, tissue derived cells, as well as histologic specimens. And so we've adopted these three different technologies: mass cytometry, imaging mass cytometry, as well as codex fusion immunofluorescence, high parameter immunofluorescence technologies. And so what we like about these, they're high dimensional. We can look at multiple different tissue specimens, and it gives us single cell resolution in tissue and with cells. And so the first technology we adopted when our course started in 2017 was mass cytometry, or CyTOF, which stands for cytometry by time of flight. And so this was developed by Fluidigm, which is currently called Standard Biotools. And so this system merges the fluidics technology of flow cytometer with ICP MS detection system. And so the inductively coupled plasma mass spectroscopy is used to detect heavy metals. And so the probes that we use are metal labeled antibodies, and most primarily in the lanthanide series. And so this and our time of flight detector can detect a range between 75 and 209 Daltons. And so that gives us 135 unique potential channels to interrogate individual cells. Realistically, we're currently looking at potentially 50 as more metals become available, this number will expand. And so one of the things that we found is there's a high technical and infrastructural barrier to implementation of this technology. And so that's why we currently have it in a core. We have dedicated operators that do that operate the machines, as well as do cell preparation. And then antibody panels are fairly easy to develop. We routinely develop antibody panels that contain 30 or more markers, and they can easily be designed. And so how this system works is you process your cells similar to how you might process samples for a flow cytometer. So you stain cells, except this time we're using metal labeled antibodies. They're introduced through a sample loader up in there in the upper left. The labeled cells are then introduced into a nebulizer, And then this nebulizer sprays the cells into a heat chamber, which drives off the water. And then these cells then enter a plasma torch where the cells are atomized. And then this generates an ion cloud. And then this ion cloud then enters into some high pass ion optics that filter out the low atomic mass particles as well as the higher atomic mass particles so that you can focus primarily on the metals that were tagged on your antibodies. And so then these enter the time of flight detector where they're detected, enumerated, and then integrated on a per cell basis to generate an FCS file similar to what you would similar to the output you would have for flow cytometry. And then the tabular data from this then can be analyzed. And so this is generally how the analysis works for CyTOF. The first step is usually a dimensionality reduction step. So we use algorithms like TISNI, UMAP, or VISNI to reduce those high dimensional data into a two-dimensional plot that you can actually visualize. And then downstream from that, we'll do clustering. And so we use algorithms like Phenograph, Full Zone, Cluster X, and then overlay that data onto the TISNI map, where we can then start to identify specific clusters based on cell marker expression, either through TISNI or a heat map. And then once you have cell classification, and then you can start to classify your cells. And then from there, then you can start to look at differences between individual groups within your study cohort. And so the follow-up technology to this is imaging mass cytometry. And so the imaging mass cytometry uses the same antibodies that we use in mass cytometry, except we're staining tissue sections. And so we can stain formalin fixed, paraffin embedded sections, as well as frozen samples. And once those tissues are stained, we can enter them, or we can put them into the Hyperion imaging module, which is in front of the mass cytometer there. And so this imaging module contains a laser ablation unit that ablates the tissue at 1 micron resolution. And so each micron then forms an ion cloud that is input into the Helios mass cytometer. And each micron then is quantified for each of the individual markers. And then you can go on from there to generate pseudocolored images based on a variety of markers in your panel. And so this is a magnified view looking at each pixel level. So an individual pixel is going to have data for all of your markers, up to 35 or more markers. And so this is just a pseudo-colored image looking at 6 different colors. This is in all of the colors within the image, but it generates for very stunning images. And so the next technology that we intend to bring on is the Akoya phenocycler fusion system. So it's the upgrade to the Codex system. And with this system, you can analyze theoretically 100 or more markers. I think realistically, maybe it's a little bit lower than that, but we'll see. And so the resolution with this is going to be slightly better than the Hyperion, because we're using imaging techniques and immunofluorescence rather than the ICP-MS and laser ablation. And so here you can also get whole regions of tissues within your sections, whereas with the Hyperion, we're looking at regions of interest that are pre-selected by an investigator. And so validated panels are currently available. However, customization can be done as well. And so how this system works is that instead of having antibodies linked to metals, we have antibodies that are linked to oligonucleotides with specific barcode sequences. And then we use fluorescent reporters that correspond to those specific tags. And so each antibody has its unique barcode. You add the fluorescent reporter image, and then you just wash off the reporter and then repeat that cycle multiple times. So you can get over 50 different markers on a single tissue slice. So this is just an example of an image that you can generate with the Codex system. Looking at those various markers, DNA, pancreatin, CD68 and CD45. But these images are great, but doesn't really leverage full capabilities of the the data, right? You have 30 or more different markers. So how do you integrate that in a way that allows you to take full advantage of that? And so we've developed a spatial imaging data analysis pipeline. I have to give a shout out to Raymond Moore, who's the senior bioinformatician that works with us at Mayo. He's in our Department of Quantitative Health Science. He's been very instrumental in developing this pipeline. And so what he's come up with is basically using a lot of open access programs. He uses a Python algorithm to export data into OME TIFF files, and then these files then are used for pixel classification in a program called Elastic. Basically, that program is used to generate a probability map for cell nuclei, cytoplasm, and background. And then that map is used then to help generate the cell segmentation map. So, and then from there, you can do classification, take advantage of your full panel to classify each of the individual cells. And then from there, then you can look at your individual cell-to-cell contacts. So this is just an example of a cell segmentation. You're going to get a nucleus, and then the outer edge is going to be your cytoplasm. And then this is your cell classification. So we use a machine learning algorithm in QPath to identify specific cell types, and then run this algorithm to identify all the specific cells in the tissue. And so this is just some of the data output that we've put together. We get cell categorization, multiple different cell types. You can map specific colors to them. Then you can start to look at cell frequencies in different tissues. But most importantly, now you can start to look at the cell-to-cell interactions. So we talk about the contact, what's the contactome? What cells are interacting with what within these tissues. And then finally, what we're finding is that there's actually these cell communities. And so there's clusters of cells of maybe 10 to 15 cells that tend to cluster together. And this can be repeated in different disease states. And you can see that they, these are often repeated, and it's kind of, and like in this particular sample, we see CD4 T cells that are interacting with multiple different APCs in and around in specific different regions in follicles, for example. And that's because you kind of see a repeated pattern. And then you can look at the frequencies of these cellular communities as well. And then finally, I just want to acknowledge the people I work with in the Corps. A lot of this wouldn't be done without all of these people here. So thank you.
Thank you so much. That was fascinating. If the speakers would like to come up, and then we'll take questions from the audience. Yes.
So I've got a question for Guy. I'll wait until he gets up the stairs.
Yeah.
So these exosomes that are released from the acinar cells, so you can detect them in the pancreatic juice. Does that mean that they're specifically released across the apical membrane, or could they also be released across the basolateral membrane? And if so, what's the function?
If so, what?
And if so, what's their function in the pancreatic juice?
Good question. First of all, they are obviously released apically. Whether or not they're released basolaterally is not clear, because we've not have a way to figure that out. Maybe under pathological conditions they are, based on the work of Herb Gazzano. What is their function? Every tissue releases these things. Some release more, What's very unique here is how robust the system is, and I mean, you don't have to wait a day. You put CCK on, 30 minutes later, you get a large amount of these things that are released, both in vitro and in vivo. I suspect, I don't know, based on your work, maybe it's got, their exosomes are known to signal to other cells, especially in cancers. It's more kind of paracrine. But it very well could be also antimicrobial. They release RNAs, siRNAs, proteins. Different cells have different complements of different types of things. So I don't have a good answer.
I mean, in the context of cancer, at least I think of exosomes as perhaps being a mechanism for sort of metastatic,
Sure.
you know.
Yeah, they can reach everything
being
from
released into the blood and circulating, and then, you know, lodging somewhere else in another tissue, and then
Yeah.
that cell can take on the various phenotypes of.
Right. They can
the
release, you know, RNAs that go into other cells that alter function. That's all starting to emerge now. Early on, exosomes were like, yeah, they come out, but nobody knows what they do. Now it's starting to emerge that they do things.
Okay, really interesting. Thank you.
Thank you.
Hi, can you hear me? Yep. So it's Kevin from University Network, the Medical Center. So the thing, like I see a lot of tissue-based analysis. At the beginning, it's a really good talk, informing a lot of information for me. So the question I'm having is like the extracellular secreted like enzyme or cytokine, they are also in the tissue, but when we do the washing and other steps, they basically losing a lot of things. Do we have method to fixing this secreted enzyme for like a extracellular matrix modification enzymes or cytokine to visualizing this kind of information from the tissue? Maybe for the panel, I think. I think the question can be like whoever have any information can just answer it. Yeah.
Can you repeat that?
So the thing is like we do a lot of tissue-based analysis. We do antibody like antibody kind of basic techniques. Then the issue is when we do the tissue-based analysis, a lot of cell secreted enzyme and cell secreted cytokines, they could be washed away. So is there any way to make sure we can also include this secreted cytokine or enzyme information in this analysis?
I guess what he's referring to is the secreted things within the tissue. You might have all that spatial information from the tissue, but you don't have the secretory component of it, which is, it's a great point, but I think it's a matter of technical barriers, right?
Yeah,
Like, once you have a tissue and it's fixed, you can't really,
Yeah,
you know,
so
how you're gonna-
I think what happens is, I mean, there are certain cytokines that are stored in cells, but there are also a lot of cytokines that are readily secreted just as soon as they're made. So I think it depends on what's being secreted. I mean, I think it's, you know, there are some cytokines I think that can be readily measured in, you know, like NK cells, for example, readily store interferon gamma, whereas T cells, you know, they have to be activated, secreted, and they secrete it immediately, so it wouldn't be associated with that cell. So yeah, I mean, I have people ask that question all the time, and they want to look at secreted things. You know, if we're doing it in vitro, like by a flow cytometry experiment, we'll have to block with like Profaldin or something
yeah,
like that
yeah,
in
that's
order
true,
to
yeah.
see it. But I don't know, you know, maybe there's techniques where you could perfuse with Profaldin or something like that. That might help to preserve that, but you know, I think just, yeah.
Okay, thank you.
We're here.
This question is for Guy. And this is with respect to RAB5 in your model for acini or acinar cells versus RAB5 in the classical model where it is an early endosomal protein on the cell surface. So in your model, you showed it coming off more from the Golgi compartments, not from the cell surface. So why so, and how do you reconcile it? This is specifically related to the question where we see class 3 PI3 kinases to be active is more in endosomal maturation, but I always imagined endosomal maturation more to be a transport from the surface down towards the cell rather than from the Golgi outwards.
Yeah, that's a great question. What I showed was anterograde endosomal trafficking. There is retrograde, which is the classic. You pull it off the cell membrane that goes into the early endosome for processing. I didn't depict that on the slide because it's complicated enough. But you're right. Rab5, though, if you localize it, it doesn't come from the plasma membrane. It stays in the early endosome. You don't see any in the plasma membrane for the most part. I'll put it to you this way. If you overexpress Rab5, you turn that system on for secretion like crazy. If you use a dominant negative, you do the opposite. So it is involved in the anterograde pathway. That's the short of it. I can give you more, but is there a second question?
Yeah. So is there a retrograde pathway then, or is it only anterograde?
Oh, God. Yeah, very robust. You have to pull all those snares and everything off the membrane. You don't make new snares for every granule. They're all recycled.
Is Fred Gorlick for Dr. Lee. I was wondering, certainly the clearing technology produces spectacular images and also new three-dimensional information. And I wondered about your experience or others here with the pancreas in particular, and if they've applied that to both murine and human pancreas, and if it's been applied to archival human tissues, and what special problems are faced and how people have circumvented those obstacles.
So I personally don't have experience, but we were our former lab were trying to establish the clarity, but yeah, I don't think I can answer those questions because I personally don't have experience. But maybe some other people can answer. Whoever has experience with the pancreatic tissue, right?
So for pancreas, remember, Bomi, the people that had done was in Sang Kim. Maybe you can say something about Sang Kim's lab. They published for diabetes specifically, and they were interested more actually on the vasculature, but they were able actually to do the clarity interestingly without having any issues, because the way this method works is immediately as you take it, you embed it or you throw it into this like for 3 days or something like that. So it keeps actually, it's almost like you're putting it in formalin. So nothing the proteins stay the same. And then slowly there is this gel that really takes, it depends what you want to look at, but they haven't had any issues, but it hasn't been tested in a pancreatitis tissue. So we were trying to do it in a mouse. I think Merle and others were trying to play a little bit with the mouse pancreas, but the normal one, not the pancreatitis. So I don't know in terms of pancreatitis, but in a normal pancreas, they've done it for human. There's actually a publication, very nice one, where you could see the vasculature or all kinds of things that it does work.
Yeah, I think what's, a little frustrating because we did it in brain tissues. You can use archival brain very easily. And I think it's just a difference in the composition, but of course we have access to all kinds of archival pancreatic tissue, but it's very difficult to get that processed, and the backgrounds tend to be very high.
So I'll tell you, here we are having this conversation. My fellow who was trying to use actually, he was looking at the intestine, and there is a learning curve. He had actually to optimize. So it didn't work. Example from Carl Deseroff's lab, he went there to do a workshop for about a week or something like that to do the intestine. But even within the workshop, he needed to really work. So I think it does require, even though there is a protocol that one follows, that needs to be optimized for the specific tissue. And I don't know about archival because we tried just to take fresh tissue and try to do it that way.
Hi there. Thank you very much for all of your presentations. And obviously that's inspiring the use of your different methods. I have a question which is perhaps very simple and maybe easy to answer for Bomi and Kevin, really. And that is with Saitov. one is getting relative quantitative information about different cell subtypes and how they might change dynamically, potentially. Is there a way in which those cell numbers can be quantified in absolute terms so that admittedly the concentrations of and amounts of different cell types of the immune system are constantly changing in the flux throughout the body and so on. But from the point of view of actually what's in the blood, can you actually quantify in absolute terms in any way? Because of course the cytoph blasts the cells completely to bits, but can they be quantified?
Yeah. Excuse me. So yeah, that tends to be a little bit difficult. I think, you know, there are people that are using like fresh whole blood and doing direct lysis, but the processing steps for cytoph tend to be a little bit more stringent than, you know, some of the full cytometry assays, and there tends to be cell loss. So I guess, you know, if you were to say that it's uniform cell loss across all cell types, you know, maybe, yeah, it could sort of be quantitative, but it is a little, you do lose cells. So it's not, I don't think it's absolute.
Might there be any way in which some form of spiking could be used that can be used in other things like RNA analyses where you can refer back to absolute quantities?
Yeah, that's actually done. That is done, but like I said, you just through the processing alone, you tend to lose cells. So I think you would always underestimate the number of cells.
Okay, well, maybe some solution for one of your future slide sections, Bomi. Thank you, Kevin.
Yep.
Sorry, I had a question, maybe completely out of ignorance. So with the spatial transcriptomics, right? You're looking at a 2D section and you're looking to looking at the cell-cell interactions or where cells are placed in relation to another. But it's in a the pancreas is a 3D, or any organ is a 3D system. And so is it that we might be relying too much on one section that you're looking at and missing something else that's important? And is it that you have to do something like a confocal where you get Z stacks or something?
So yeah, there have been attempts to do this on a three-dimensional basis, but it's a little bit more difficult. But yeah, I agree. I think three-dimensional data is better. But I think, you know, what we have right now is two-dimensional data, and that's sort of, I hope we get there, but you know, we're sort of stuck with two-dimensional right now. I think, yeah, maybe more, you know, if you're doing confocal or something like that, you'll be able to get that, but not with this technique.
Actually, Tamil, can I ask you a question? So when it comes to the scoring, T1 scoring that you're developing, because you're trying to validate that, right, in these big consortiums, for the time of implementation, if it gets implemented for other radiologists, do you think there's gonna be some pushback because it takes too long? Or do you think like radiologists will like come on board and just do it and implement it? Can you talk about that?
Yeah, absolutely. Thank you for a great question. Actually, I will detail about this tomorrow about the T1 score, which will be an abstract presentation. The T1 signal intensity ratio or T1 score is a very simple imaging method. It is already being used in most of the MRI imaging, and it does not require contrast, and it takes only 20 seconds to acquire. So it's very simple and practical MR imaging tool that gives information about the entire pancreas, not only just one slice, and it reflects the pancreatic exocrine function and also correlates with the histopathology, and more details tomorrow morning.
Thank you so much. Well, this was an excellent session. If there are no other questions, I think we had some great discussions and we've learned a lot we can then close this session and give you guys 4 more minutes back. Take a break. Thank you so much.